let's go ahead and get started. Welcome Black. I hope everyone had a nice little break on Tuesday. Maybe spend some time catching up on their molecular biology. So yeah, just a reminder, please use the class feedback survey when you get a chance. We will have homework posted tonight, and if you need motivation to do that homework and hand it in, it is very clear that in general, people who are handing it in for points have about a 10-point higher median midterm score than those not doing it or rarely doing it. So there's still time. There's still homework 6, 7, 8, 3 more homework posts for which you can get extra credit and assess how well you are understanding the material to know where your strong and weak points are. All right, so let's review what we talked about in the last class. So our last class was focused on post-transcriptional control of gene expression. What that means is after you have transcribed that RNA, it's gotten processed in the nucleus, it's been spliced, polyadenylated, and capped, there are a lot of things that can happen to it that will affect how much protein it can be translated into, as well as even the sequence or type of protein. We can change the coding sequence of a messenger RNA. And that was the first thing we talked about, these modifications that can happen to the messenger RNA itself. We call this generally RNA editing. You can change the base so that it'll become another. That could change the codon sequence, which could change the amino acid, or even make it encode then for a stop codon to make a truncated protein. We also talked about an enzyme called Adar. Adar converts adenosines into what base? Adenosines become inosine, exactly. So A to I. When you have an inosine in a messenger RNA, it is read by the translation machinery as a G. So if you see an I in the messenger RNA, because it has been deaminated from A to I, just treat it as a G if you are decoding it. We then talked about this general property of mRNA stability. At some point, all RNAs are going to be degraded, but they can get degraded at very different rates, depending on sequences or elements they may have that might stimulate deadenylation or decapping, or even cause cleavage in the middle, endonucleolytic cleavage. And that's actually a process we'll talk a bit about today when we talk about RNA interference. We then learned that there are surveillance systems to make sure that the proper messenger RNA is what makes it to the translation machinery. If there are splicing mistakes in the nucleus, those are usually recognized there. The RNA is trash, never even makes it out of the nucleus. We talked about a special situation where sometimes we can end up with a premature stop codon, so a nucleotide change that changes a codon into a stop codon that is before the natural stop codon. And we talked about one mechanism that is able to recognize those nonsense mistakes and get rid of that RNA. What was that mechanism called? There is chomping involved to get rid of if you have a premature nonsense codon. Yes? It is. So it's nonsense-mediated decay. Nonsense is another word for a stop codon. So you have a premature stop codon. You're going to decay that messenger RNA. And one of the mechanisms by which that stop codon can be recognized is through this exon junction complex. So whenever your messenger RNAs undergo splicing and those exons are ligated together, the last step of that splicing is there is a complex of proteins that are deposited on that spliced messenger RNA. We call this the exon junction complex. They're there at every junction. And this complex of proteins stays with the spliced messenger RNA out into the cytoplasm until the first round of translation. Translation, ribosome comes through, knocks off all those exon junction complexes, and that's good because the exon junction complex, what does it recruit to an RNA that it's stuck to? This is where your chomping comes in from the 3' end. deadenylases, right? So you have a poly-A tail, so you deadenylate. So there's deadenylases that get recruited by the exon junction complex. Then a normal messenger RNA, ribosomes knocked off all those exon junction complexes. But if you have a premature stop codon and the ribosomes fall off before knocking off all the downstream exon junction complexes, they stay stuck to the messenger RNA. They recruit the deadenylases, you degrade that messenger RNA. And so that's a mechanism, this nonsense-mediated decay, to get rid of RNAs that have these premature nonsense or stop codons. We then talked about a variety of mechanisms by which we can control the translation of a messenger RNA. First, we talked about you can globally regulate. So your cell under a certain condition, maybe you're deprived of nutrients, so you don't want to spend a lot of energy making a lot of proteins, so you can globally down-regulate the efficiency of translation. And one of the major factors that can respond to nutrient availability is this TOR kinase. And we didn't talk about all the steps that are involved with TOR kinase regulation, but basically when TOR kinase is active, when there's a lot of nutrients, it is going to promote translation. You're going to have high levels of translation. Low levels of nutrients, TOR is inactive, you have low levels of translation. And that relates to almost all the messenger RNAs in the cell, and that's what we mean by global regulation. We then talked about a few other specific ways that we can regulate particular RNAs for being translated. First, we talked about this mechanism of cytoplasmic polyadenylation. And so this is in certain cell types. We talked about in an egg as well as in certain neurons. There can be messenger RNAs that are stored in a dormant form, meaning they are stable, but they're not being translated. And the way that they are kept stable is that they have an element or a sequence in their 3' UTR called the CPE, which stands for Cytoplasmic Polyadenylation Element. This is bound by an RNA binding protein called CPEB, Cytoplasmic Polyadenylation Element Binding Protein. When it is bound to these messenger RNAs, it recruits a protein, in eggs it's called maskin, that then sort of hides the 5' end of the messenger RNA so that it can't be recognized by the normal initiation factors. And this will hold those RNAs in the dormant state until there is a signal. For example, in a fertilized egg, there's progesterone. This turns on a kinase signaling pathway. You get phosphorylation of CPEB. Now this protein kicks off maskin. It recruits cytoplasmic polyadenylation factors, lengthens that poly-A tail. You recruit poly-A binding proteins. They interact with the initiation factors of the 5' end, and you get very efficient translation of messenger RNAs that were already there. And you just needed this signal to switch the regulation and get very rapid translation of those messenger RNAs. And then finally, we talked about how iron homeostasis can be regulated by 2 different genes. One of them, transferrin receptor, brings iron into the cell. Ferritin is the sponge. It soaks up excess iron. And they are both regulated by having elements or sequences that form a structure in their RNA that are recognized and bound by an iron response element binding protein. And the key here is that RNA binding protein binds to iron itself. When it is bound to iron, can it bind to RNA? No, I'm saying no. Exactly. So you just have to remember, this protein either is bound to RNA or to iron, but cannot be bound to both. And in that way, it's able to regulate these 2 different messenger RNAs at the translational and RNA stability levels to make sure you have a balance of iron inside the cell as it needs. And then finally, the last topic, which we'll come to in a few slides, is this idea of localized messenger RNAs so you can get localized protein production. Before we move on to that, let's do a quick clicker question and answer which of the following statements about post-transcriptional control of mRNA expression is correct. 10 more seconds. All right, final answers. Okay. All right, let's go through each one. See if one of these is correct or none of these is correct. So let's just start with the first one. So A says highly transcribed genes always produce high amounts of protein. Does anyone like that or not like that? Want to explain why? Kalia.
I didn't choose that answer because might be transcribed, but maybe it's not a functional gene.
Well,
it is transcription, but that does not necessarily mean there's protein production.
Exactly. I mean, that was one of the whole points of the last lecture, right? You can have high amounts of synthesis, but if it's bound by CEBP and in a dormant state, you are not making any protein. Some RNAs are very highly transcribed, but they have those AU-rich sequences in their 3' UTR. They recruit RNA binding proteins that cause destabilization. So you might have a high level of transcription, high level of destabilization. You're not going to get a high level of protein production. So the concentration or the amount of protein you make in the cell is going to be based on how much the gene is transcribed, how much RNA do you have, and then how stable and how much is actually going to get translated into protein. So A is not correct. Any questions about that? What about B? If you know the sequence of a mature messenger RNA, you can deduce the sequence of the DNA from which it was transcribed. Arya?
No, because of the fact that a mature mRNA has to go through a lot of processing to get to the mature state, so you can't necessarily deduce the exact DNA which is transcribed.
Yeah, so what would be missing in the mature messenger RNA? Yeah, so the introns, you wouldn't have that information anymore. Anyone think of something else that can happen to the messenger RNA that would make the sequence different from what the encoded gene was? Yes? Yes, exactly. So we could have editing. You can change the sequence of the base, and you'd see the messenger RNA as that sequence, but that could be different from what the DNA was. So B is not correct. What about C? A premature stop codon in the 5' UTR will induce nonsense-mediated decay, or NMD. What do we think about that? Yes? Yes, exactly. There can be no stop codon in the untranslated region. You can't stop until you start. Once you start, then only then are you in the reading frame where you could hit one of those 3 different stop codons. So there is no such thing as a stop codon in a 5' untranslated region. Does that make sense? Question?
That also applies if there's no stop codon in the 3'.
Right, because by definition, the 3' untranslated region is you've stopped translating. Exactly. So C is not correct. Any other questions about that one? All right. What about D? The IREs, iron response elements in the 5' UTR of ferritin mRNA positively regulate translation initiation by binding to IREBP. What do we think of that one? Antonio.
It actually prevents
Exactly. So, does ferritin have its IREs in the 5' UTR? Yes. No. That is correct. So, ferritin, remember the F and 5, ferritin 5' UTR. So that is where they are. There's those little stem loops that are bound by this RNA binding protein when it's not bound to iron. But when the protein is bound there, what happens to translation? It blocks. It actually negatively regulates translation initiation. The scanning, the pre-initiation complex can't get through when the protein is bound to those IREs. So this one is incorrect because it would negatively regulate translation initiation. Everyone remember that? So none were correct. Any questions about any of these? All right, so the last part about post-transcriptional regulation that we need to discuss is subcellular localization of a messenger RNA. So what do we mean by that? We know that cells come in many shapes and sizes, and so for example, you can have neurons that have synapses where they contact and interact with other neurons. And what this figure is showing you here is that they have tagged a protein that's known to localize to synapses. And that's just saying, all right, we know in this cell, here we have 2 synapses. What these scientists also did is that they used a technique that used a probe that would base pair to this censorin messenger RNA. And when they threw that probe on the cells, whoa, it also is localized to these synapses. The messenger RNA is at these synapses. This is an example of a localized messenger RNA, and the reason for having localized messenger RNAs in the cell is so that you can get local protein synthesis. If you're going to get local protein synthesis, what else has to be in these synapses besides just the RNA? The ribosomes, the translation machinery, exactly. And so this is true for many cell types and many specific messenger RNAs. But if you looked in this cell, if you probe for a lot of different other messenger RNAs, you wouldn't find them localized here. They would be in various parts of the cell. So there needs to be a sequence or an element in these RNAs that directs them to that part of the cell. It interacts with a protein that takes them to that part of the cell. We sometimes call these RNA zip codes, just like you need a zip code on a letter to get it to the right place. That's what we call these localization sequences. So here's another example. So myoblasts, these will become muscle cells, and they need to sort of stretch out and line up with each other. And so one of the ways they get these shapes is by having actin localized at their edges. And so then the question is, how does actin localize to these parts of the cell? So suppose you know this is the sequence of beta-actin messenger RNA, the mature messenger RNA. What kind of experiment would you do to determine which sequences in this actin mRNA are doing the work, are acting as the zip code to be recognized by a protein to be taken to these edges of the myoblast? Can anyone think of a general type of experiment you could do? A reporter gene, exactly. It's a very similar question as when we wanted to know, is a gene expressed somewhere? Here we want to know, is there a sequence in this RNA that can do something? And so you could build your reporters. So remember we said the UTRs are really hot spots for regulatory elements. So that's where I would start with and trying to figure out where the zip code was in this actin RNA. Just like when we talked about reporter experiments before, you can fuse that test sequence to something that's easy to detect. It could be GFP. In this particular experiment, it's beta-gal, which we know makes X-gal turn blue wherever the protein is expressed. And so maybe you want to test the 5' UTR of actin versus the 3' UTR of actin. So these are the same, except for you are testing the 2 different portions of that actin gene. You put a gene encoding this, a reporter, into the cells. You let the RNA be expressed. It gets translated into protein. You add your X-gal, and you look at where was the protein produced. It's sort of an indirect readout of where the RNA was, but it's easier than looking for the RNA first. So when we put the 5' UTR onto beta-gal, do we get localized expression? No. So it doesn't look like the sequences in the 5' UTR are sufficient to direct localized protein expression. What about then the 3' UTR is used? Yeah, it looks exactly how we saw the whole beta-actin gene itself. So what that's telling us is that there are sequences in this 3' UTR that are sufficient to direct the RNA to a specific location in the cell and drive expression in that position. All right, so we know the whole 3' UTR can do it. How would we find the specific sequence in the 3' UTR? Yeah, deletion or mutagenesis. So just like we did with our reporter genes when we wanted to find those activating and repressor sequences, we just changed them. We looked when expression changed. Here, we would change them, and if we mutated a site and suddenly it starts looking like this, that's the sequence that was necessary to drive local translation. Here's another question. If I put this same reporter that can drive local expression in these myoblasts into a different cell type, neuroblasts, would I get the same result? Maybe, yeah, exactly. What would it depend on? What would have to be the same in those two cell types? The proteins that are recognizing that sequence or similar sets of proteins would need to be there in order to do the activity. The reporter's the same. The RNA sequence, this reporter gene, would be exactly the same I'm putting into those cells. But if I saw a different result, that means that cell type isn't expressing the same protein needed to localize that messenger RNA and allow for expression. Any questions about this? All right, so let's move on to our new topic for tonight. So we're going to talk about regulation of gene expression by non-coding RNAs, small non-coding RNAs. And we'll go through the mechanisms, actually the discovery and the mechanisms of RNAi or RNA interference, as well as microRNAs. And these two mechanisms are still sort of relatively newly discovered in the past couple of decades. And so I'll take you through sort of a brief history of how they were discovered, because they were both big surprises, and it really shows how just curiosity-driven science can lead to actually Nobel Prize winning discoveries, because they have huge relevance for human health and therapeutics, and we'll talk a little bit about that as well. So RNA interference was first discovered through studies in the model worms, C. elegans worms. These are these like little millimeter long, mostly transparent worms, not the kind of worms you would go fishing with. But these are common model animal. And this story actually begins with a graduate student, Sue Guo. She was studying this gene in worms that she thought was pretty important for embryogenesis. And when you're in the laboratory and you're trying to figure out the function of a gene, usually what you want to do is inhibit the expression of that gene, and do you see a phenotype? If it was needed, if it was important, you'll see some change. And so one of the technologies available to her in the early 90s was what we called antisense interference. And so it was well known that if you designed an RNA that could base pair strongly to the 5' end of a messenger RNA, to the 5' UTR, this could block translation, and you could get reduced levels of expression. And this is great because if you knew the sequence, it was easy to design an antisense RNA that should be able to base pair. So Sue designed an antisense RNA to block her gene of interest, and that was great because it seemed to block expression of her protein, and she got dead embryos. So, okay, this gene is probably pretty important for embryogenesis. The problem was she was a very good scientist and did an important control. She also treated a set of worms with RNA she made that was the same sequence as the 5' end of her RNA. So it was sense. Should this be able to base pair and block the ribosome? No. There's no way we know of base pairing where sense pairs with sense. So this shouldn't have been happening, but she got the same phenotype. And so, oh no, what is going on here? And so that was very troubling, but luckily for Sue, she actually managed to isolate a genetic mutant of her gene, show that indeed it was very important for embryogenesis. She published this big fancy Cell paper, and in the methods, she included this experiment even though she didn't understand the outcome of it. And that was good because that caught the attention of other scientists who were like, wait, this is really weird. And then some other people were able to reproduce that like sense RNA is causing inhibition. And so when you get strange, reproducible results like this, you need to take a step back and think about, all right, how actually was that experiment done? What went into it and what might be happening that we hadn't considered? All right, so if you're setting up this experiment, remember the first thing we need to do is we need to make an RNA that will base pair to the 5' end of your RNA. So you know what sequence you want to make, and we can make this in vitro in a test tube. We know how to make RNA in a test tube. If I want to synthesize a specific RNA in a test tube, what do I need? What's one of the things I need? You need a template, yeah. So you would design a template that has a promoter, and then we'll encode the sequence of RNA that you want to make. So you have your template. What else do you throw in that tube? Yeah, NTPs, nucleotide triphosphates, the building blocks to make that RNA. And then what do you need to do all the work? Yeah, proteins, the polymerase complex. And fortunately, we actually know viral polymerases that you don't need all those general transcription factors for. And so in the laboratory, you just need this simple viral polymerase with its promoter, and it'll pump out tons of this RNA for you. All right, so you make the RNA, and then the way this worked in C. elegans is you can actually inject that RNA with a needle into the C. elegans germline. These worms are hermaphrodites. That RNA gets taken up by the germ cells, put into the embryos to interfere with that gene that you targeted, and then you can ask, is there a phenotype? And in her case, yep, you got dead embryos, okay. And then the control, of course, was almost exactly the same, except for you make sense RNA. And so what you can just do is take your promoter, flip it on the other side, so now you will make RNA in the other direction. You've designed it. It will not be able to base pair to the gene you're interested in. Take that, inject into the animal, and this is where the problem came. You get this phenotype that was exactly the same as the antisense inhibition. And so really some very clever and careful work by Andy Fire and Craig Mello came up with the solution of what was actually happening in this experiment. Turns out that whenever you are making this RNA in vitro, you're throwing in tons of template, tons of polymerase, and a very small percentage of the time, the polymerase goes on the wrong side, even though you don't have a promoter, and makes a small amount of the other strand in the same tube where you're making a lot of the RNA that you wanted to. In other words, you're making a small amount of double-stranded RNA whenever you do this in vitro transcription. And they went on to show that if you purified the sense away and purified the antisense away, actually neither one of those gave very good knockdown phenotypes, even though we knew about this antisense technology. But if you specifically mixed them together and injected the worms with double-stranded RNA, you got this very potent knockdown phenotype. And so this discovery of double-stranded RNA causing RNA interference, or RNAi, is what won them the Nobel Prize almost 10 years ago. But you're probably thinking to yourself, okay, if sense RNA can't base pair to a target sequence, how in the world is double-stranded RNA going to base pair and interfere? And so this was the work of a lot of different labs once this key piece of information came out. And we now know the basic mechanism of what happens when a cell is introduced to long double-stranded RNA. So DS stands for double-stranded RNA. So this is the double-stranded RNA that was being injected into those worms. We now know that our cells have an enzyme called dicer. Dicer is a specific RNase. What it does is it will cleave that double-stranded RNA into about 22 nucleotide long with little overhangs on the 3' ends that we call small, because they're small, interfering RNAs, or siRNAs. And these are what work in this RNA interference process. All right, so you have double-stranded siRNAs. What happens next is there is a protein called Argonaute that will peel these strands apart and take up 1/2. so a single-stranded siRNA. And then this complex of the Argonaute protein with its siRNA guide and also some other accessory proteins, we sometimes call RISC, for RNA-induced silencing complex. So what this does is the single-stranded siRNA that is part of this complex goes around and tries to base pair. Once it is perfectly base paired to a target sequence, this triggers the Argonaute protein in RISC to chop the target messenger RNA in half. So you get endonucleolytic cleavage of the target. We saw last week what happens if you cut an RNA in half. You have no protection on either side here. There are going to be exonucleases that are then going to degrade this RNA in the 3' to 5' direction, and this RNA from the 5' to 3' direction. It is a very efficient and quick way to degrade an RNA, take it out of commission, knock out its gene expression. One important thing to notice here is that the guide, this siRNA, it does not get cleaved in this reaction. Only the target messenger RNA. What that means is once the target messenger RNA has been cleaved, this RISC complex can go and find another target, cause cleavage. Go and find another target, cause cleavage. So you need a very small concentration of these siRNAs in order to do the RNA interference. And this is why when there was that contaminating small amount of double-stranded RNA in those experiments, still worked very well because this is a catalytic mechanism. All right, so this is great, and it was shown to work very potently in C. elegans, also Drosophila cells, and some other models. But there seemed to be a problem if we wanted to use RNAi in mammalian and some other types of vertebrate cells. And that is because it was well known that these types of cells, when they see double-stranded, relatively long double-stranded RNAs that are 30 nucleotides or longer, this triggers a general immune response. Your cells sort of recognize this as, ooh, there might be a double-stranded RNA virus around. We better shut down gene expression of everything. So this seemed to be a problem because this would be non-specific inhibition of gene expression, right? It wouldn't matter what sequence your double-stranded RNA is. Everything in the cell is going to be decreased in expression. So can you think of a way, keeping in mind the mechanism I showed you on the last slide, how do you think RNAi could be used to still inhibit specific genes in mammalian cells? So if you're bigger than 30 nucleotides, you're going to cause all kinds of general inhibition. So what could you do? Yes? So 30 nucleotides and longer will all cause the inhibition. So go the other way. Yes, exactly. So you can just start with already these 22 nucleotide siRNAs. This is the substrate that Argonaute needs to see to pick up and then make this RISC complex and go around and find the targets. And in fact, this is what we do in mammalian cells. Antonio?
Would you treat it with the dicer in vitro, or would you just build it that way?
Yeah, so there's it is cheaper to do it with dicer in vitro if you don't mind having many different siRNAs against your gene, and that can be very useful. But there's also many companies now where you can just order your siRNAs that you design to be any sequence that you want. But either way, as long as you are already down to this size, that immune surveillance isn't going to see this short of small RNAs. Yes, Claire? So yeah, so that's a great question we are going to be leading up to. So worms and other organisms that don't have as sophisticated an immune system as us do indeed use this to defend against viruses that they get infected with. Our immune system has a different layer and usually isn't dependent on dicer in this system immediately to defend against the viral RNAs of this size, but it definitely is a viral defense in plants and things lower than mammals and most vertebrates. So what this means then is, yes, Madeline?
So then if it's bigger than 30
So there are proteins in the cell that are kind of sensors of double-stranded RNA, and they only can sense them when they're 30 nucleotides or longer. And most viruses, if they come into the cell, would be of that length. If you put in a 22 nucleotide, those sensors don't see it. They're too short. They don't bind to that size, and therefore they can be picked up by Argonaute and go into this pathway. For us to use as an experimental method in the lab is essentially what I'm coming to for this. So one of the important discoveries of RNA interference is that we now have a mechanism where we can knock down expression of any gene we want just by knowing its sequence, right? It's super easy to design this siRNA. It just needs to be able, one of the strands has to base pair to your target of interest. And it's a very potent mechanism in most cell types. Most cell types have plenty of Argonaute, take up these siRNAs, knock down your gene of interest. We just said that you can directly use siRNAs. That's what we use in mammalian cells. You can buy them from companies. In fact, we now have what we call RNAi libraries. What we mean by the library is we have siRNAs to target every single gene in the human genome. So you could do a screen if you wanted to say what genes are essential for these stem cells to develop into myoblasts. You can just take populations of cells and treat with every siRNA against every gene in the human genome and look, when do I not get the cells being able to develop into that cell type? When do I see the phenotype? And then you already know what the gene is, right? Because you know what siRNA you use for that. So this has been a very powerful method to do screening and discover functions for genes that we had no idea what they did before. We have these, of course, in C. elegans as well, and we're actually lucky in worms. Worms are amazing. They actually eat bacteria, and you can put vectors in bacteria to express double-stranded RNA against every gene in the worm genome. The worms eat it, they take up that double-stranded RNA, and it goes into the RNAi pathway. So we don't even need to inject them anymore. It's a very powerful RNA knockdown method. So what I'm showing you here, for example, is a transgenic worm. So it is expressing GFP and a variety of cell types. You take this same exact strain of worm, it has this DNA transgene in it, and you put it on bacteria that are expressing double-stranded RNA against GFP. 24 hours later, no GFP. It's all been knocked down. They still have the gene, they're still transcribing that GFP, but RNAi is knocking down the messenger RNA before protein can be made. And so this idea that you can pretty easily design siRNAs to target any dangerous gene that you want, like a viral protein or many human disease genes, has led to many different companies starting and developing therapies based on this RNAi technology. There's now a handful that have been FDA approved, and they are actually being used in human patients with good efficacy. The biggest challenge, like many drugs, is delivery. So remember, it's easy to design the siRNA. If you have a protein like this mutant form of transthyretin, so this is a mutant form of this forms sort of aggregates in your liver, it travels in your body, you get neuropathy, it's like really terrible. But if you knock down the expression of it, you stop making those aggregates, and it can really help patients who have this mutant form of the gene. But that means we need to be able to deliver the siRNAs to the liver, which makes this mutant form of the gene. Fortunately, everything you eat, every drug you take goes immediately to your liver automatically, right? And so most of these, almost all of these FDA-approved siRNA therapies are targeting liver genes. But of course, there's many other diseases that we would like to target with this siRNA technology. And one of the current hopes is by using what are called lipid nanoparticles. And so the name is exactly what it suggests. So you have the siRNAs enveloped with lipids. The lipids not only protect the siRNA from being degraded in your bloodstream or digestive system or however they're delivered, But sometimes we can actually fuse peptides or other conjugated molecules onto these lipid particles that help route them to specific tissues in your body. And so there's a lot of interest in trying to direct these LNPs with their siRNA cargo to the tissue that needs it, where you want to have this kind of knockdown. All right, let's do a clicker question. So to the right are results of an RNAi experiment to compare the percent of cells undergoing cell death in 2 different cell types exposed to siRNAs against BCL2 gene. Which of the following could explain the results? So whenever we do RNAi against a gene we're interested in, we always do what's called a control, where the siRNA is designed to not be able to target anything in the human genome, just to make sure the way you deliver to the cell is not causing the phenotype. And then, this is working well because we're not getting any phenotype with our control siRNA. But then when we are actually targeting BCL2, what's happening here? Ten more seconds. Final answers? It's got to be more than one of the above, right? That's a good, safe answer. Let's see. All right, so let's start with A then. Alternative splicing of BCL. Well, first, let's just make sure we're all on the same page on how to interpret some of these data. So we can see if we look at our fibroblasts, we put the control siRNA in, and there's no cell death. We put the RNAi again in against BCL2, 95% of the cells are dying. So what is the normal function of BCL2? What would you interpret from that at a very basic level? Yes? It's preventing cell death, right? So when you don't have it, when you knock it down, the cells all die. So basically, that's how you would interpret that. So A says alternative splicing of BCL2 in hepatocytes reduces its targeting by the BCL2 siRNA. So we look at hepatocytes. What's happening here? Ooh, big difference from fibroblasts. So what do we think about A? So first, how is our RNAi experiment going in the hepatocytes compared to fibroblasts, for example? Antonio? not much is happening. It's almost exactly the same as the control siRNA. So we're not getting really a phenotype, so maybe our gene is not being targeted. Could that be happening if you have alternative splicing in hepatocytes that's different in fibroblasts? Yes, because why? Exactly. So if you designed your siRNA to target a particular exon, and that exon gets removed, but you still make a functional protein, it just might be regulated different, then RNAi is never going to see it if your mature RNA does not have the target site in it anymore. So that is potentially an explanation for why we are not getting a phenotype and potentially not having RNAi happen in that cell type. So A is a possibility. Does everyone see that? You need to have the target sequence in order to have an RNAi effect. Excuse me. B says BCL2 protein is very stable in hepatocytes. Could that explain our hepatocyte results? Sure.
No, because if it's stable, that means it's promoting cell death.
So remember, it's the opposite. So we said that the job of BCL2 must be to prevent cell death, because when we knocked it down strongly here, we had a lot of cell death. So then you change your answer. Yeah, so if we have a lot of BCL2, it's already protein. Does RNAi target the protein? No, it targets the messenger RNA. If you already have a ton of protein and that's a long-lived protein, you might not see an effect of your RNAi experiment. Maybe if you let the experiment go on for many, many doublings of the cell, you'll eventually get a phenotype. But the point here is just to emphasize that the protein is not the target of RNAi. If you already have the protein, doesn't matter, right? Does that make sense? All right, so B is a possibility. What about C? BCL2 is reduced equally by RNAi in each cell type. So what do we mean by that? It means the level of the mRNA is equally reduced. How would I test that? Northern blot to test the messenger RNA. What if I want to test protein? Western blot, exactly. So I could test that I'm getting a knockdown of this gene equally in both cell types. All right, so I did those experiments and oh, yeah, it's knocked down compared to the control. But the cell death machinery is regulated differently in fibroblasts versus hepatocytes. Is that possible? Maybe hepatocytes, you don't need BCL2 to prevent cell death? Sure, maybe there's a BCL1 in there. Maybe there's a homolog that is not targeted by your siRNA, and it takes over the job just fine. So you can have different pathways in different cell types that could lead to different phenotypes, even though you are equally knocking down the same gene. Does that make sense? So C is a possibility. What about D? Hepatocytes have lower levels of Argonaute than fibroblasts. If you have lower levels of Argonaute, what's going to happen? You're going to have less RNAi, exactly. And this, in fact, is true. There are some cell types that have very low levels of the Argonaute you need for RNAi. Doesn't matter how much siRNA you put in there. If there's no Argonaute to do the actual work of finding the target and cleaving it, you're not going to get an RNAi effect. So D is also potentially an explanation. So it was actually all of the above. Any questions about that? Yes?
So, Professor, 95% means that only 5% have cells undergo cell death?
Yeah, so we're looking at the percent of cells. So we have a population of cells. Let's say we have 100 cells. Of course, it would be more than 100 cells. Almost all of them are dead. So we are getting a very strong phenotype compared to our set of cells where we've used a control siRNA where none are dead.
So how come then B is a possible answer?
So in hepatocytes, we have almost no difference from the control. Only 1 out of those 100 cells is dead. Right? So if we have a lot of this BCL2 protein, it's still there protecting almost all those cells. So 95 to 1 is a very big difference, right?
So this siRNA is destabilizing the BCL2?
The messenger RNA.
Yeah.
The siRNAs would target the messenger RNA. So maybe the messenger RNA is being targeted just fine in hepatocytes, but these hepatocytes already have a ton of BCL2 protein present to protect the cells. And the point here is that RNAi can't do anything to a protein once you already have the protein. It targets the messenger RNA. Does that make sense? Antonio?
So just to clarify, it's the protein levels that are stable, or is it the structure of the protein?
Could be either, as long as you have enough of it to protect the cells there. Yes, Nick?
So the Argonaute is guiding the siRNA?
Other way around. The siRNA is guiding the Argonaute. And because by guide, what we mean is it is what finds the target. And how does it find it? Base pairing, exactly. So the siRNA is already in a complex with Argonaute, and it's going around sampling. Can I base pair here? Can I base pair here? Whoop, I have strong base pairing. The Argonaute protein is then there, cleaves through the messenger RNA. Any other questions? All right, so then this kind of comes back to a question that came up previously. So why do our cells have this RNAi machinery? I mean, it's great. It gives us a really powerful tool to use in the laboratory, to use potentially for therapeutics. I told you in mammalian, in our own cells, it's not really used so much for viral protection because we have this more sophisticated immune system. But it turns out that these proteins, this dicer and this Argonaute, are actually part of a natural pathway that is present in all of our cells and very important for gene regulation. And this is the microRNA pathway. So microRNAs are a type of small regulatory gene. So your genome encodes these microRNAs. They are encoded and expressed differently in all your various different cell types. Just like messenger RNAs get transcribed as a pre-messenger RNA and undergo processing to make the mature messenger RNA, you'll see that microRNAs are initially transcribed as much longer RNAs that also undergo processing to produce the final functional. They're usually around 21 or 22 nucleotides long. That's where their name comes from, micro, tiny. And generally what they do is they inhibit expression of protein coding genes of messenger RNAs by base pairing to the target mRNA sequences, usually in the 3' untranslated region. So how are microRNAs discovered? Well, so they are also discovered in C. elegans. So it's not just that I love C. elegans, it's just that both of these small regulatory pathways were discovered in worms. And the microRNA story actually came about through just very curiosity-driven science, trying to understand how this animal develops. And so, as you're all aware, our genes are encoded in our DNA, and if we have a mutation or a change in a gene, that can get copied into the RNA that gets transcribed from it. If it's a messenger RNA, that can then get translated into a different type of protein. And if that protein was important for a function in the organism, you then might see a phenotype by having defective expression or levels of that protein. And so there was a very interesting worm that had a phenotype, and this worm was called Lin4. Lin stands for lineage. They have abnormal cell lineages. What the scientists could see is that these worms would actually go through normal embryogenesis. They'd hatch into a first larval stage worm. But then several of the cell types, like the skin, the intestine, some neuronal remodeling, would get stuck in that early larval type fate, while other cells in the body would continue to develop to become an adult-sized worm. Now, I mentioned before, these worms are hermaphrodites. They are self-fertilizing hermaphrodites. What that means is they produce their own sperm, they produce their own oocytes, they come together inside of the animal. The animal produces an embryo, an egg, and normally lays it outside of the body. One of the cell types that is messed up in these Lin4 mutants are the cells that are needed to lay those embryos outside of the mother. So these Lin4 mutants, normal germline, they still make progeny. There's no way for those progeny to get outside of the mother. So all these little squiggles you're looking at inside the worm are baby worms eating their way out of the mother. So really horrible fate for the animal, but really easy phenotype to notice as geneticists. And it immediately tells you whatever this Lin4 gene is really important, right? And so with that idea, of course, the initial idea is it must be some really important transcription factors because they do everything regulating expression. And that's why it was really quite shocking when Victor Ambros was able to go in, identify the mutation in the genome, and see that the only gene product that was actually produced from that mutated region was a tiny RNA of about 21 nucleotides long. Definitely cannot encode a protein, right? So this tiny RNA. And then fortunately, around the same time that Victor was discovering what Lin4 was, he was good friends with Gary Ruvkin, who was studying another gene that was important for cell lineage. This was called Lin14. It was known that Lin14 encodes a protein. And what they realized together is that there were sequences in the 3' UTR of this Lin14 messenger RNA that could partially base pair to this Lin4 RNA. And the genetics had already told them that Lin4 regulates the expression of Lin14, and that's how they were able to put together this new mechanism for gene regulation. So Lin14 has its protein coding region, and in its 3' untranslated region, there are actually multiple positions that can partially base pair to the Lin4 RNA. So when we talked about RNAi and those siRNAs, we designed them to perfectly base pair to the target. And when they do cause cleavage of that messenger RNA degradation. Turns out that none of these sites can perfectly base pair to the Lin4 small RNA, and this is true for almost all microRNAs in their target sites. And there's not even an overly consistent sort of base pairing format, so this makes it really hard to identify target sites by microRNAs. But through a lot of genetics and molecular biology, Ambros and Ruvkin were able to establish this completely unheard of before form of gene regulation where the organism actually produces this tiny RNA. This tiny RNA finds target sequences and shuts down expression of that protein. And in fact, that is what was needed so that all those cell types could stop being early larval stage, turn off this early larval stage protein, and then develop on to the later stages and become normal adult animals. And so this all was discovered in, published in about 1993, and it was pretty cool and interesting, but nobody really cared about it because this protein coding gene, Lin14, there was no sense that there was a homolog of it outside of worms. There was no evidence that these types of RNAs were present in any other organism, so it was just kind of thought to be, well, a cool worm oddity. And that was until about 7 years later when additional work in Gary Ruvkin's lab was again just studying a worm that had a really interesting phenotype. These worms actually will develop normally up until about the last larval stage before they're going to become adults. And then they actually rupture. They explode from their midsections. So this stuff is all the guts and gonads spilling out of the worm. LET stands for lethal. I think you can appreciate how this is a lethal phenotype. So again, this must be a really important developmental gene. When they went down and discovered what it actually was only a single nucleotide change in a sequence that they found encodes another tiny RNA gene. And then additional work in Gary Ruvkin's lab found that there is a protein coding gene, this time called Lin41. I know it's confusing. Don't memorize the names of these proteins. That's not important. But it also has sequences in its 3' UTR that in this case, there's 2 sites that can partially base pair to this LET7 small RNA. And when LET7 comes on, it represses the expression of Lin41. That allowed the animal to differentiate cells to make them stable so that the worms do not explode like that horrible phenotype I just showed you. And then what was really exciting, so this was in 1999, and so large parts of the Human Genome Project started to be published online, freely available. And so when we took the sequence of the LET7 small RNA, so this is just the C. elegans genome, and we took this sequence and searched the human Homo sapiens genome, we found an exact match. So that was really exciting, 21 out of 21 potential, but a genome that's 3.4 billion base pairs long, that could just happen by chance. And so what we really wanted to know is it possible that humans might actually be expressing this type of LET7 RNA? So what experiment did I need to do to see if LET7 was actually being produced in human cells? A northern blot, exactly. And that's what I did. And so here in the first lane, I included an important control. So I knew this RNA was produced in C. elegans worms. So this lane has human RNA. And then I got cadaver RNA from all these different tissues and ran that on my northern blot. So we all remember how to do a northern blot. We take our RNA, denature it, run it through a denaturing gel. We transfer it onto nitrocellulose or a stable sort of membrane, fix it, design a probe that will base pair to your sequence of interest, and then detect. And so what you can see is that in all these different human tissues, there's different levels of RNAs that had to be the same or at least almost the same sequence of LET7 in order to base pair to my probe. I stripped the blot, reprobed for a housekeeping gene, ribosomal RNA, so you can see pretty standard levels in all these tissues. And so this was interesting because now I can conclude there are different levels of LET7 produced in these various human tissues. Remember I told you that we could not find a sequence that matched Lin4 in the human genome? And indeed, that still is true. And if you make a probe that is complementary to the worm Lin4 and you reprobe this same blot, you see no expression of RNAs matching that sequence in the human genome. And so this was the first indication that it's not a weird worm thing. Humans and probably all animals in between actually encode these types of small RNAs that then generally became known as microRNAs for their small size. And we now know that there are hundreds of different microRNA genes in all multicellular organisms that we know of. Question? Lydia? So there are some targets that we know about in worms that actually are conserved in humans. In humans, there are actually a lot more targets and more tissues, and it plays an important role. So you saw that actually, even though the levels are quite different in various tissues, there's sort of a lot of LET7 in almost every tissue here. That's not true for many of the other microRNAs that we've discovered since. There's some that are only made in the brain or only made in the heart. And so LET7 turns out to have a very broad role in cellular differentiation. We'll come back to that in a moment when we talk about some examples of what happens when you don't have enough LET7. Our own human genome is thought to encode over 1,000 different microRNA genes. What I mean by that is the mature sequence is different, therefore potentially different targets. And so this is very complicated. We are far from understanding all the different genes and all the different pathways that are regulated by these different microRNA genes. What we do know quite a bit about is their basic molecular mechanisms of how they are expressed and how they do function. So the general microRNA pathway is outlined here. So remember, these microRNAs are genes. They are encoded in your genome, just like your protein coding genes, just like your messenger RNAs. So they need to be transcribed. Most of them are transcribed by your RNA Polymerase II complex. So remember when we talked about regulation of messenger RNA gene expression, there are all those activators and repressors and chromatin involved? That's all going to hold true also for microRNA genes. And that's going to be the reason why Some cells only express one type of microRNA. Other cells express a different microRNA. Depends on their genes and their promoter elements. The initial transcript that is produced from the microRNA gene isn't 22 nucleotides. It's much longer, often hundreds to thousands of nucleotides long. And that initial transcript that's produced is called a primary microRNA, so the first microRNA. While it's still in the nucleus, this microRNA undergoes a processing step by this Drosha enzyme complex. It is going to find the hairpin that includes the eventual mature microRNA, and what it does is it clips that hairpin out. So you're just left with this shorter, about 70 nucleotide RNA that we then call the precursor or the pre-microRNA. This pre-microRNA then travels out to the cytoplasm. This is then where it is recognized by the dicer complex. So remember, dicer was that enzyme that takes our long double-stranded RNAs and chops them up into siRNAs. So the precursor has a lot of double-stranded RNA nature, so dicer is able to recognize it. It chops off the loop structure. We temporarily have these double-stranded mature microRNAs, and then one strand is chosen to be bound by Argonaute and become part of this RNA-induced silencing complex. The mature microRNA, just like an siRNA, is then what serves as the guide to try and find the target to which it can partially base pair. So in red here is the microRNA, and these sequences here and flipped out here is just to indicate that there is not perfect base pairing. There's some level of partial base pairing. It is recruited Argonaute protein. And then Argonaute is sort of a scaffold that can recruit a whole bunch of other proteins. And through complicated mechanisms that are not completely understood, this usually results in the target messenger RNA being repressed for translation and being targeted for deadenylation and mRNA degradation. Any question about those steps? Yes?
So is it the loop of the hairpin or the straight part that becomes the part that we're going to be using?
The mature in the end? So yeah, so here is the precursor that comes out to the cytoplasm. Dicer, shown here with the two scissors. Loop gets chopped off, that gets degraded. What you have left is that duplex right there. And then one strand is going to be peeled away and put into this RISC complex. So in this case, it's this 5' end, this UGG. So this strand is what got bound here. Yeah, so that's a good question. Most microRNAs are very asymmetric, meaning one strand is really preferred as the mature form. And there's some thermodynamic rules that guide which strand is more easily peeled off and put into Argonaute. The strand that does not get taken up by Argonaute, exactly, it's unstable, it gets degraded very quickly. Question, Claire?
Just going back, so in less complex species, just like RNC and Argonaute systems use trials defense, but then
So the microRNA pathway is present in plants as well as probably all multicellular organisms. And organisms that don't have the more complex immune system that we have also use dicer and sometimes Argonaute to defend against viruses. Things that have more complex immune systems just have a different pathway. But all of them that have microRNAs will use dicer and Argonaute for the microRNA pathway. And then this is a really cool RNAi and microRNA animation. If you like to visually watch things happening, I highly recommend you check that out. All right, so coming back to the question we had before. So does LET7 do the same thing in worms that it does in humans? Well, to a very broad sense, yes, it promotes cellular differentiation. And by understanding what the function of microRNAs are, in particular, the genes that we know are targets that they regulate, We also can see what happens in many different diseases or cancers. The levels of certain microRNAs change. If the microRNA level changes, if the microRNA level goes down, targets are going to go up. That can cause the disease phenotype. So here's an example with LET7. So in most of our differentiated tissues, as I showed you with that northern blot, you have very high levels of mature LET7 microRNA. And that's important because some of its targets are genes that might ring a bell to you: MYC, RAS, HMGA2, cyclin D2. Does that ring a bell, any of those proteins or genes? What are they often involved in? Cancers. Yeah, they promote, so these are normal genes your cells have, but they promote cell division. And there are certain cell types or times in development where you want to have a lot of cell division. Most of your differentiated tissues, you do not want those cells to be dividing. So you have high levels of LET7 to keep the levels of those cell division promoting genes down. What happens in a cancer? Many cancers end up having low levels of LET7. A mutation disrupts the production of the LET7. It targets it for degradation. Some way you get low levels of LET7. What does that mean? Now you're going to get weak repression of all these cell division promoting genes. They're in overdrive. They're going to tell those cells to start dividing. Then you get a tumor. So LET7 very often is thought to be a tumor suppressor because it is suppressing these cell division promoting genes. So by knowing the microRNA and its targets, you can start to think about how we can develop therapeutics to either add back or in some cases actually suppress a microRNA that is overexpressed and could be potentially causing the disease phenotype. And so there's a lot of companies and different therapeutics that are being developed. When we add a microRNA back, for example, in those tumor cells, if we want to get LET7 back, we call that adding a mimic. So we just design a LET7 and try to develop a delivery system to get it where it needs to go. In some cases, it is actually overexpression of a microRNA that is causing the disease phenotype. So to deal with those overexpressed microRNAs, we can design a small RNA that will base pair to the microRNA itself, taking it out of commission. We call those anti-mirs. And so there's, again, a lot of interest in these types of therapeutics because we know the sequence of the microRNA. It's very easy to make a mimic or to make an anti-mir. But packaging it and getting it into the cell type where it needs to function is still a pretty big challenge. And so far we've talked about siRNAs. These are the small RNAs that function in RNAi. MicroRNAs, that's what we just talked about. And unfortunately, we're not going to have a chance to talk about all the many other types of non-coding RNAs that we now know play very important roles in our own biology. So some of you may have heard of piRNAs that are very important for reproduction and fertility, maintaining our transposons. There's a whole set of what we call just generally long non-coding RNAs. So microRNAs, siRNAs, piRNAs, they tend to be short, about 20 nucleotides. Things that are longer, we generally call long non-coding RNAs. There's thousands of examples of these types of genes in our genome, many of which we have no idea what their function is. And then the last type of sort of RNA-guided gene regulatory mechanism that we'll talk about is those directed by CRISPR. And so that's sort of the lead-in to where we will start next Tuesday in talking about genome editing using CRISPR.